A cell has a membrane, which is like a plastic bag that keeps the contents of the cell together. Those contents include all sorts of complicated machinery for making proteins, for example, and they include also the DNA and the RNA and all the rest of it for turning genes into specific proteins, and then for recycling those proteins, building them, all that sort of stuff. And lots else besides needs to be done by the cell, and all of that is held together by the cell membrane. The cell membrane is made of fat, and that fat is a double layer, and it contains chunks of stuff embedded in it. And those chunks are receptors. Now, a receptor is a protein, but it's a very complicated structure. So if you look at them 3D, they look like sort of swirls of knitting wool or something. They're very, very complicated. And they are embedded in the cell membrane. Now, you get various types of receptors because there are lots of different kinds of neurotransmitters and neuromodulators in the brain, and they all have their own particular kinds of receptors. But you can classify the receptors into two kinds: ion channels and metabotrophic receptors. Basically, ion channels are fast, metabotrophic receptors are slow. Together, those ion channels and metabotrophic receptors provide a very complicated way of regulating the cell's activity, because they can incorporate both fast information and much slower changes, and those slow changes may include changing genes so that some genes are activated, i.e. producing protein, and some genes are not activated, i.e. not producing protein. So you can use that on many different timescales, both to change the cell's electrical activity, but also to change what genes are expressing proteins, which in turn changes what's going on in the rest of the cell. So ion channels and metabotrophic receptors provide complicated, multi-layered, if you like, control of cell activity. The action potential is an electrical signal. What does that actually mean? Let's look at that in a little more detail. You have this fat, this membrane. Now, fat is an insulator, so it is possible to have an electrical difference between the inside and the outside of the cell. So you can have charged atoms and molecules. Charged atoms obviously are ions, and you can have those at different concentrations inside the cell than outside, and that means that you can have what's called a potential difference, a difference in electrical charge inside and outside, and that is the basis of everything that you are and everything that you do, because you rely on that difference to exist, to think, to feel. So, when you have an electrical difference, and at rest a neuron has an electrical difference of about minus 70 millivolts, which is called the resting potential, which means that the interior of the cell is more negative than the exterior. When you have that, what happens when an action potential comes along is that that very rapidly becomes a lot more positive. How does it do that? Well, it relies on these fast receptor types that I've talked about, called ion channels. An ion channel, if you think of it like a donut, only a donut with a hole, it sits in the membrane, and that hole can be closed or open depending on what state that complicated three-dimensional receptor structure is in. What does that depend on? Well, it depends on what other chemicals are in the environment, because that receptor is specialised to link up with one particular kind of chemical, which might be a neurotransmitter like glutamate or GABA or noradrenaline or whatever, or it might be something more specialised like a passing hormone, or it might be a neuromodulator, which these are chemicals that are released sort of alongside neurotransmitters and kind of have a more modulatory role. They don't generate a signal, but they might affect how it's processed. So you have this ion channel, and this ion channel has a particular chemical called its ligand, from the Latin word ligare, to bind, and that chemical, when it comes in, it will form a chemical bond with the receptor and it will change its shape, and that opens the channel. What happens when that channel opens? Well, particular ions, charged particles, can get in through the channel, or indeed sometimes out through the channel, depending on what kind of channel it is. When you get an action potential, the ion channels that open are sodium ion channels. Sodium is a very small, positively charged ion, and it floods into the cell. There is quite a lot of sodium outside the cell, there's not so much inside, and so the sodium floods in and the electrical balance rapidly becomes much more positive because suddenly the interior is becoming much more positive than it was previously. That all happens in almost an instant, in less than a couple of milliseconds. What happens then is that triggers other ion channels to open. Those ion channels are potassium. Now, potassium is another small positively charged particle, but there's lots more of it inside the cell than there is outside. So out they go, and because you've got positive charges leaving, down comes the electrical potential on the negative side, and what you get is a slight overshoot as it goes slightly too far, and then it rebalances back to the resting potential. And that is how an action potential is generated, and all of that takes a few milliseconds, that's all. It's very, very fast for the cell to generate this spike, recover itself, and then it's got specialised mechanisms called transporters which pump all the ions back to where they were to begin with, and then it's ready to do another one of these things, perhaps in as little as 3 to 5 milliseconds. All of that is spiked, done, dealt with, reprocessed, transported back, and you're ready to start again. So that's an action potential. Ion channels control the fast electrical changes which happen on the cell membrane. Metabotrophic receptors are slightly different. They're much slower, and they control what happens in the innards of the cell. A metabotrophic receptor is attached not to a hole, not to an ion channel, but to a lump of stuff inside the cell called a G protein. And so what that means is that when the particular ligand that activates the metabotrophic receptor comes along, that receptor changes shape, but instead of opening a hole in the membrane, it changes the shape of the G protein, and that triggers a whole lot of intracellular cascades of activity that will then lead to, for example, certain genes being switched on that weren't on previously, certain proteins being made that weren't made previously, and so on. So metabotrophic receptors are much slower. Now, it all gets rather complicated because you can have the same neurotransmitter, such as glutamate, activating multiple receptor types at the same time. So the glutamate may be released into a synapse, and it may go across and it may activate something called an NMDA receptor, which is a particular kind of ion channel on the recipient cell, and that will trigger a fast effect on that cell. It may also activate other ion channels called amphokinase receptors on that same cell, so you've got two different sets of activity going on just in the cell membrane. And as well as that, the glutamate is also perfectly capable of activating metabotrophic receptors, which are going to do much slower changes. So you've got complicated control just from this one neurotransmitter. And it gets even more complicated than that, because you can also have that glutamate feeding back to receptors on the cell that released the glutamate, and quite often you'll get a sort of negative feedback cell, because although I've talked about receptors as triggering changes in the cell, not all of those changes necessarily lead to the cell being more active. Sometimes they can damp it down, they can inhibit it. So you can have receptors that are what they call heteroreceptors, i.e. they activate another cell, the Greek word hetero meaning other, or you can have autoreceptors, which activate the same cell that released the stuff, from the Greek word for the same, autos. You can have all of those, and you can also have excitatory receptors, which make the cell more likely to signal, and inhibitory receptors, which make the cell less likely to signal. So it all gets rather complicated. And then if you add in upregulation and downregulation as well, then it gets even more complicated, but that's another story, and I'll deal with that elsewhere.